hold on. I'm going to give you a brief helping explanation of the graphing, the cell transport graphing assessment that most of you are going to go straight to that and just do that now. Because a lot of you have already done the retake, and if you've already done the retake, you're ready to go on to the cell transport graph. So this is a helpful, really short explanation of that, the cell transport graph assessment that is right now, it's right at the top of your Google Classroom. Okay, so they're going to explain an experiment to you where, and I'm going to draw for you because a picture always helps. So they're going to show you a graph of data, but they're not going to really, they don't give you a visual. They just give you a description. I'm giving you a visual. So once again, we've got a beaker with a solution in it. And floating in that solution, there is a plant cell this time, okay? So that there is a plant cell, and you know plant cells have cell walls and membranes, and they have cell membranes that can control what goes in and out of them. Okay. So then they're going to give you a table, and it's going to be two things. Okay? Yeah, this is an individual exercise. That's why I want to give you the explanation, so when you get there, you'll understand what you need to do. Okay, so on one side, it's going to be concentration of glucose, right? That's going to be the concentration of glucose is in the beaker water out here, right? So the concentration of glucose in this water out here is what's in this column here. And the units are important. The units are in millimoles. You don't need to worry about what a millimole is. Millimoles per liter. Right? So amount of glucose per liter of water. Okay, so for example, the first one they're going to give you is 0, which is the control. And then the next concentration of glucose in that water is going to increase to 10, and then the scientist increased it to 20, and so forth. Okay, I'm just going to give you those first 3. So this out here, the amount of glucose that's in the solution around the cell is what the scientist is manipulating, right? Okay, so remember that. Remember what that would be. Would that be the IV or the DV? Think about that. Okay, then the next column is the absorption of glucose into the cell. Okay, and that is a speed, and it's in units of moles per gram per minute. Okay. So that's going to be the data that they're going to give you. I'm just going to give you just a few of those data so you can kind of look at this. When there's no concentration of glucose around the cell, it's 0. When there's 10 millimoles per liter around the cell, the speed of the glucose going inside is 300. It goes up really fast. And then at 20, it's like 400 or something like that. Okay, so let me explain this. So out here, for example, when the concentration of glucose out here is 0, then the rate, right, the speed at which the cell is absorbing the glucose is 0. Right, so these numbers describe the amount of glucose in the water. Maddie, are you having an issue? Are you all right, hon? Oh, okay. Oh, I think your mic is off, Mr. Bouchard.
I said they're upset because I said no teams.
Oh yeah, well this isn't. Well, you have a long time to do this, so don't be nervous about it, guys. You're going to get a lot of help with this. I'm just trying to give you an idea of what the experiment is about before you go in and you're faced with all this. You don't have to do any calculations here. You're just going to create a graph using the table and then write a claim evidence reasoning, right? So yeah, I know you can't see my mouse, but that's why I'm making drawings. Okay, Luke? So you'll make a graph and then you'll do a claim evidence reasoning. But if you don't really know what's going on with a picture, with a visual, I think it's harder to do, which is why I'm drawing this for you, right? So the amount of glucose is what's on the left. So the amount of glucose on outside the cell, right? The amount of glucose outside the cell is what this first column is telling you, right? So if it's 0, for example, then the speed or the rate, right? The speed the cell absorbs glucose is this arrow here, right? Right? So if there's no glucose in the water around the cell, then the speed at which the cell is taking it in is also 0. That makes sense, right? And then look what happens as you increase it. So if you increase the concentration outside the cell to 10 millimoles of glucose there per liter, then the speed increases to what? What does the speed increase to? If you have 10 millimoles of glucose around the cell, outside the cell, yeah, it increases to 300. So this arrow over here, when it's 10, it increases to 300 millimoles of glucose per minute is going in, right? So it's much faster when it goes up to 10. And then when it goes up to 20, look what happens. And then the data increases after that, right? So you're going to make a graph that shows the IV and DV here. I don't want to give too much away, but you're going to make a graph. You're going to look at the graph. You're going to make sure the graph has a good title. What's a good way to do a good title, guys? How do we always write the titles? Let's review that for a second. Graph title. What's the skeleton of a good graph title? We always say effect. Yeah, there you go.
Effect IV and DV, yes.
Whatever the IV is on whatever the DV is. That's it. Right? So that goes at the top of your graph once you're done making it. And then when you label your axes, make sure you include the units, right? Just copy what it says there next to each, the IV and the DV. It's right there on the data. Just copy what it says. And then remember, your claim is just, it's going to guide you right through what to write for your claim evidence reasoning. It tells you exactly what to do, all right? But this is the situation. The scientist is manipulating the amount of glucose outside the cell, right? The amount of glucose out here. So that's what the scientist is manipulating. And then the scientist is measuring how fast the glucose gets absorbed into the cell as she increases how much glucose is around it, okay? So that's basically the lab in a nutshell, and then you have to take it from there. Let me show you exactly where it is. It's at the top of your Google Classroom. I don't want to erase this. I think I want to keep this here. I don't want to erase this. I'm just going to keep it up. I like it. But it's at the top of your Google Classroom, and there's a copy for each of you. Okay, so I'm going to stop recording this. You should have enough now to do a good job on this. This isn't due until the 22nd, but if you get it done now, then it's off your back. Especially, it's a good thing to do it now because we're right here and we can help you. So definitely take advantage of that and get as much as you can done today. Unless, of course, you still need to do a retake. If you need to do a retake, go and take care of that first.